about this super variant, the lineage B117. So, for our listeners, this speak of a new variant, a new set of mutations that is more transmissible, and potentially more of everything, virulent, etc., has been, shoved down our throats in the last month. And it's all come from the UK, come from, the COG UK initiative of bringing Lighthouse Lab genomic sequencing together to see if we can find both mutations and if there are dominant variants. So I've written about this just a little, pulled together some resources. But what are your thoughts on this? And maybe the first question is, and this is, tinfoil hat type question, so not meant to be, but are you confident, Claire, that the work earlier in the year, in January, to fully isolate SARS-CoV-2 and therefore fully sequence it, so we have a full kind of signature of that virus, do you believe that that work has been done and it is irrefutable? As a result, therefore, we can do genomic sequencing, noticing we've got the full picture of what this virus is?
I do. I think there was a virus, a new virus. I think we've done lots of work on real patients done by Public Health England, as well as others, where swabs from real patients have been used to infect cells in culture, and they replicate and they explode the cells. And we've managed to do a whole genome sequencing of it, which is where you read every single letter of the nucleic acids. So you read the whole recipe for the virus. And, I think that we have got enough reads of whole genome sequencing that's really covering the whole genome of COVID, and it's repeatedly the same.
Without human elements to it as well, right? We're confident that is like a fully isolated genomic sequence.
Yeah, I am.
Okay.
I think it's real. The thing is, whole genome sequencing is a new test, and it's been used in medicine for a few years, and it's been used as a way of getting additional information. So a patient where you already know what's going on in terms of diagnosis, but you'd like extra information about anything else you can find, then you might get a whole genome. But it's being used now as a quantitative test to say whether or not somebody has a diagnosis. And it's being used for mass population screening. Well, not as a primary test, but for confirmation. And when you have testing that's being used in that way, you have to be really careful, and you have to understand how and how often it can go wrong. And you can only understand that by really stress testing it and almost trying to make it go wrong. You sort of stop and you think, well, how could we be mistaken? And that work really hasn't been done. And they're just slowly starting to do a bit of work around that. But I fear that on whole genome sequence reports where we shouldn't have been confident that it was definitely there, it will have been reported as being definitely there. And the thing with the UK is that actually, thanks to things like the 100,000 Genomes Project, we have amazing capacity for genome sequencing. So we have done a lot more of it than other countries. So it's not surprising that we'd be the ones talking about finding variants, because we're looking for them more. But what we found with this particular variant is that it's been around since September, and it's just been kind of gradually, slowly pootling away, maybe increasing a little bit. And then in mid-November, it went from that gradual pootle to something much, much more dramatic. And looking at that dramatic change in the proportion of cases, apparently with this variant, looks to me like a laboratory error. When you see things that suddenly change and suddenly do something very dramatic, your first response shouldn't be to go and run in a hole and panic everybody that we're all going to die. The first rational response to that is to think something's gone wrong and to see what could have gone wrong. And my first thought about what could have gone wrong that would make the South East, the East Midlands and London have a sudden increase in this variant is that somebody in the lab may have been shedding RNA over the samples. And I'd said this in a speculative way, but then of course we found out the 24th of December that there'd been an outbreak in the Milton Keynes lab, and 20 out of 70 of the staff are off now. So I'm not sure that was such a crazy suggestion. I think that is what may have happened there. And it's not until, things settle down, give it a couple more weeks, we'll find out whether that was the case, because they won't suddenly be quite as many of these variant as they apparently were.
That's a very interesting hypothesis. I did see you state that on Twitter. I mean, I guess the counter argument would be, okay, it was moving slowly from at least, the samples that we took. It wasn't as prevalent in September and October, November, into December, things start to pick up. Someone would say, well, that's just, that's an exponential curve. That's the quote unquote, the R number, doing its work as it's working through the population. Yeah. What do you say to that?
Well, I don't buy that it appears everywhere at the same time. So I don't know quite the sort of rationale behind this in terms of which lab is doing the testing. But the other thing that happened in mid-November was that this new variant appeared in every pocket, like every corner of Wales, which, Wales is not a country that people visit every corner of very often. And for it to all emerge on the same day across Wales just looks like a reporting error. Like that's not how infectious diseases spread.
Okay. And, the error could be, as you say, could be contamination, but it could be a change in process as well, right? Potentially that, something to do with the sequencing. I don't really know how sequencing works, but I guess there could be updates in the AI and the kind of testing process, and because a lot of this is mathematical, isn't it? Sequencing.
Yes, and so one of the problems that we now have is that because they've diagnosed this new variant, they're saying that the tests that did require 3 genes to be positive on PCR now should only require 2, because the third one might not go positive with this new variant. So suddenly the criteria for testing has changed to make it way more easy to get a positive.
No way.
So, that's happened, guess when? Just before we've suddenly seen this massive uptick in the number of cases.
When you say testing, you're talking about PCR testing?
I am, yeah.
Right, okay. So there's less, is it primers or probes? There's less nucleic acids to test for. They only test for 2 instead of 3. Is that what you're saying?
That's essentially it. So effectively, if you're testing for 3 genes, it's almost like doing 3 different tests. So if you had, say, a 5% positive rate, a false positive rate for 1 gene, but you're testing for 2, then you have 5% of 5% as your false positive rate. You test for 3, you have 5% of 5% of 5%. The more times you test, as long as you require all of them to be positive for you to say that it's a positive test. But if you then remove one of your tests, then you've just multiplied that up by 5%. Wow. You're going backwards.
And now we're seeing huge numbers, right? 50-odd thousand reported cases, the highest we've ever seen, making spring just look like a nothing in comparison to what we're seeing now. Now, I know that the response will be, but we were doing selective, targeted testing without the capacity and without the wider community approach. We're now, scaling our testing regime, and it's the scaling of it that's, finding the true prevalence that we weren't able to identify previously. I don't really buy that, to be honest. But
I think that was true. That was true in May. So in May, in April, we didn't have sufficient. We don't think we had any real community testing outside of care homes. So in April, we were not seeing the true number of cases. By May, we started to do quite a lot of testing, and by June, we had 100,000 tests a day. That's tons. So, I don't think that argument could be used subsequently. That was an argument for then.
Yeah.
But on the 50,000 cases a day, that's registered cases. If you look at the cases by specimen date, it was very much a pre-Christmas peak, and it's turned and it's coming down. And I think there were a lot of people who went and got tested before seeing Granny for Christmas.
Yeah.
I'm I'm not saying they should have seen Granny for Christmas, just I think that's what may have happened.
Just as we kind of move off of this variant thing, is there anything else you want to add? I mean, from my perspective, in my research of this, that mutations are very frequent and to be expected. That I know there are tens, if not hundreds of thousands of reported mutations on the SARS-CoV-2 virus, and there are thousands upon thousands of identified variants and many lineages. So it's not surprising that we're talking about a variant. It's easy science to kind of cherry pick from to create a straw man that we have an issue. However, in your point, from your perspective, do you think it's fair and it's right and it's appropriate to be talking about the variants of the virus? Is this a red herring, or is it viable that we pay attention to the phenotypes of these variants that we're identifying and coding?
I think we should be paying attention to phenotypes, but I think this is a total red herring. So I think you're right. There's been tens of thousands of variants, and they'll continue to be. And the wonderful thing about deadly viruses is they always get less deadly. That's how things evolve. They become more spready and less deadly over time. That's historically what viruses do. And there's so many people working so hard to try and find any evidence that this is more transmissible or more deadly or less deadly. There's tons of people working on that. So don't worry, we'll know if it ever has any effect. Thus far, no evidence.
Yeah, I wonder how long this variant discussion is going to go on for. I feel it was just it was the criterion which governments could say, look, our hands are tied. We have to do something. We have to do something new. We're going to have to lock you down. And then once we've got something else that we can blame or something else that we can praise, that we'll forget about the variant discussion. And I suspect we're going to probably see that in the coming weeks, especially as there's further push on the vaccines. Talking about the testing stuff, we've spoken about. You've mentioned PCR testing, lateral flow testing. I know, case definition is driven only by PCR testing, confirmed cases, that is. So PCR is kind of held up as the gold standard.